Hi, I'm Svati Shah. I'm a cardiologist at Duke University and former chair of the American Heart Association Genomic and Precision Medicine Committee and chair of the American Heart Association Research Committee. I'm joined today by Drs. Andrew Landstrom and Anwar Chahal, who led the writing group which drafted consensus-based guideline for interpreting incidental variants. Andrew, what sorts of variants are most often flagged, and why are some reported while others are not?
So as Anwar mentioned, there are particular guidances that are in place from the American College of Medical Genetics that suggest that certain variants in certain genes should be reported back. And this can be in the research setting, or it could be an exome or a genome setting conducted in the clinical arena. And so predominantly these are variants that are being reported back or being discussed as being things that might need to be returned back to the individual who was sequenced. This list is not inclusive, and the benefit of oftentimes this kind of genetic testing is there's lots of knowledge, specialized knowledge among the individuals who are doing the gene testing, and so this list may grow depending on the perspective of the research team or the clinical team. But as Anwar mentioned, this actually creates the complexity that we see because there is no set guidance for what to do with those variants, for how to actually interpret them, whether they're on that ACMG list or not. And what do you do when you're stuck with the findings? And it basically boils down to planning ahead. So before you get that clinical exome, before you get that research-based exome, have you considered the possibility of finding an incidentally identified variant? How have you built that into your thinking, your research pipeline? And those questions about what kind of variants will you report back? What kinds of gene variants will you report back? Should then start to in the discussion if you're having that plan. And we found that oftentimes people can gain a lot of traction when you have the variants, if you've anticipated what those possible things could be before you ever got the gene test. So one aspect of this I think is critically important.